Right, thanks. My name is Jill Goldthwait, and I'm the Director of Government Relations for the Jackson Laboratory. And I appreciate the opportunity to be here to present two requests for federal funding. The first is entitled Human Response to Infectious Disease, and this request would provide one component of a project that we're working on at the laboratory to provide a type of laboratory space that we do not have now, and that is called Biosafety Level 2 or BSL2 space. BSL2 space can be used for low-level infectious organisms, the kind of thing you might be exposed to in a doctor's office waiting room, for instance. And it would provide the capacity for our scientists to do some types of research that they are limited in doing now because we have jury-rigged a small amount of space, but basically we don't have any BSL2 functional space. We are now building an importation facility on campus. It will be online in the fall of this year. We have enclosed the third floor of that building as shell space and are now looking to fund the interior construction, though not through this request, and also equipment for that space so that it is fully functional and ready for our scientists. We have scientists who have programs that are actively seeking this type of space. We would be funding the construction portion of this request, which is about $1.5 million. This funding request is for half of the equipment. So we'd be funding construction and half of the equipment. This would fund the other half of the equipment. One of the kinds of experiments that would be done there has to do with taking a mouse called a humanized mouse, which actually contains human tissue engrafted into the mouse. So when you're working with that animal, you are not working on mouse tissue, you're working on actual human tissue in a living organism. So you can see the response of that immune system tissue, for instance, in a living mammal. So it's a great research opportunity, and you can do work in that, frankly, that you cannot do experimentally in humans. So that's the basis for this request. One of the ways that that mouse is used is you need a viral vector to carry information into a cell. And so it's not that we're infecting mice with infectious disease there, but we are using viruses to work with that humanized mouse to explore the mechanisms of infection in human beings and understand how you could interrupt those pathways. It has both civilian and military applications, obviously, because the goal is to understand how an organism gets infected and what you can do to interrupt that infectious process.
Great.
All right, let's take a breather and then we'll do the